Please welcome to the stage Dr. Paul Sheehan.
Good afternoon. Welcome back. So, in the last pandemic, it is hard to say that we were in any way lucky, but we did catch one break, right? We had seen coronaviruses before, like SARS and MERS, and that really sped up our ability to do diagnostics and provide countermeasures. We might not be so lucky the next time. So part of the Friend or Foe program's goal is to find unknown pathogens that have never been seen before, quickly identify how they might infect, whether they infect, and give them a score for pathogenicity, right? So how do you play this, how do you find unknown bacteria? And the real goal is to play what I call the world's fastest and smallest game of 20 questions. You isolate bacteria from the environment, you query them individually. Hey, does it attack a membrane? How does it defend itself? Does it have antibiotic resistance? Does it bind within the host, right? And all together, you pull that together for a ultimate score for pathogenicity, and then that allows you to identify bacteria that may want to study a little bit more, right? That might be future human pathogens. So to that end, we've had several teams. My favorite team was right here at Texas A&M, led by Professor Rumhan. He is the Texas Instruments Professor of Electrical Engineering, and they have developed, with colleagues, Paul DeFigueroa and Jim Samuels, the iSentry system. Arum, over to you.
So the US military operates globally in an extremely diverse environment. So does our civilian population. During all of these activities, they come in close contact with the natural environment, including both soil and water. So microorganisms exist in all of these environments with extremely high diversity and numbers. Unfortunately, some of these microorganisms are harmful and can cause major disruption to our military and civilian operation. So how do we detect these bacterial pathogens? One way to overcome, the current gold standard is DNA analysis, in other words, genotyping. They are widely accessible, they're reliable, and they're fast. However, they require prior knowledge. This means that they cannot detect unknown pathogens. So how do we overcome this challenge? One way is to measure the functionality, in other words, phenotype of this bacteria. However, the method of phenotyping has not changed much in the past century. They remain low throughput, they are time-consuming, and because of these, they can provide only incomplete coverage. So one way to think about this problem is asking many questions to these microbes of interest. So they could be, for example, do you kill a human cell? Do you attach to a human cell? Are you resistant to antibiotic? Are you able to evade detection of by the human body, etc.? Importantly, in all of these, because of the extremely high diversity and number of microbes, being able to do so against a large number of microbes very fast is important. So you can think of this as the world's fastest game of 20 questions with a vast repertoire of bacteria, one cell at a time. So if you were to use the conventional technology approaches to answer this question, it becomes impossible. So what you're seeing here on the top is lots and lots of plates in which bacterial cells are growing. This is something you see in a typical microbiology lab. Our approach is lab on a chip, in which we can reduce all of this complex instrument and assay into a very small form factor to answer these complex questions. So what does this chip do? Our droplet microfluidics approach utilizes millions of picoliter volume bioreactor. These bioreactor sizes are smaller than the diameter of a human hair, where each of these droplet bioreactor can encapsulate single cells and then be interrogated extremely fast. In the past decade, genotyping, so obtaining genetic information from cells, have rapidly grown in terms of their throughput. However, phenotyping, in other words, analyzing the functions, has not been. So the goal of our iSentry system is to rapidly advance the field of single-cell phenotyping as well as bacterial theta assessment. The iSentry system is an integrated microfluidics-enabled system for phenotypic detection of biothreat agents. So here we show an overview of our iCenter system. It has 3 components. The first component is extracting microbes from an environmental sample. Second, single-cell phenotyping on this extracted bacteria microorganism, followed by analysis and validation to determine whether a microbe of interest is a phrenophore. So let's look at the first extraction process. We have developed a soil microbe extraction technology that we were able to now reduce into a very simple, small kit format to allow us to harvest highly diverse, highly viable bacterial cells from soil sample. On the right side, you are seeing the successfully extracted bacterial cells that are highly viable. So these extracted bacterial samples now get encapsulated into a droplet at a single cell resolution at a very high speed. So these millions of droplets are then incubated to amplify the encapsulated microbes so that downstream phenotyping becomes possible. So compared to the current state of the art, we can recover more total cells, more viable cells, while being able to do so thousands of times faster. So combined with a 4-fold increase in the diversity in an extracted microbe, we can really isolate and characterize more of the dark microbiome than ever before. So let's look at the single-cell phenotyping step. So as I mentioned at the beginning, trying to determine whether a particular bacterial cell is a potential pathogen or not cannot be easily answered by measuring a single function. So we have tested a variety of different assay types and configuration, and ultimately settled on 3 questions to make phrenophore determination. The first one is whether a microbe adheres to a host cell. The second one is whether a microbe causes cell death in a host cell. The third one is whether the microbe exhibits antimicrobial resistance. So let's look at the first assay, adherence assay. So in this assay, microbes are co-incubated with a human host cells. We are utilizing a microfluidic system that can enable apply electric field-based cell separation. So on the right side, this enables us to only harvest and isolate bacterial cells that are adherent to host cell, and that's what you're seeing on the right side, a pathogen that is adhering to the host cell successfully separating. The second assay is a toxicity assay. So here, microbes are placed together with a human cell in a droplet, co-incubated, and any droplet that show bacterial killing of a human host cell can then be detected optically and sorted at very high speed. And the third assay is antimicrobial resistance assay. In this assay, here microbes are cultured in the presence of a variety of different antimicrobial. These droplet-containing cells then can be dispensed on an agar plate. So the microbes they are recovering from these plates are demonstrating antimicrobial resistance and hence a potential pathogen. So this single-cell phenotyping droplet microfluidic system automates to reduce the labor. They are a closed system to minimize the chance of exposure and risk to the operator. And each of the assay only uses a millionth of a volume than a conventional assay and can do so thousands of times faster. So the final part of the iCenter component is the analysis and validation step, where the iCenter data, together with the metadata, gets uploaded to the LabKit server for easy data organization and visualization. Finally, we are making a phrenophore threat assessment based on the microfluidic single-cell phenotyping data and with the help of a machine learning algorithm to make that assessment. So our system went through several rounds of independent verification and validation, where a team from the National Institutes of Standard and Technology harvested agricultural soil, sterilized it, spiked it with more than 100 different microbes, both pathogen, non-pathogens. And so these samples went through our iCenter platform pipeline. And the key outcome here is that we are able to correctly classify close to 90% of the pathogens present in this soil sample. So the iCenter system has now been miniaturized into a form factor so that we can deploy this one inside a biosafety cabinet so that the entire operation can occur inside a biosafety level 3 lab. So the transition to CDC and FDA is ongoing. Their team has visited our lab, and our team also visited, for example, the CDC high containment laboratory in preparation of this transition of this technology. Obviously, a task of this complexity requires a multidisciplinary team, where success has been enabled by a close collaboration between Texas A&M University, University of Virginia, University of Oklahoma, UCSF, Argonne National Lab, and NHS, and as well as NIST that provided us these IVNV sample and testing. Thank you.